Okay, good morning. I think in the interest of the time, I think we will start the session. Good morning. Good morning to all participants. Good morning to Clindamines webinar on advanced pharmacovigilance. Uh, the objective of this webinar today is to uh, create the awareness or share the information about the advanced pharmacovigilance. This program, this webinar is more useful for the professionals who are already in pharmacovigilance space, the professionals who have already spent last 2, 3, 4 years into the case processing or into the other areas of pharmacovigilance, but uh, the program would cover today the information about starting from medical review, aggregate report writing, signal detection, risk management, and all the essential documents in pharmacovigilance. The objective is to create awareness, what skill sets you require in advanced pharmacovigilance, and how we could help you. In order to do that, we have amongst us Mr. Gaurav Grover. Gaurav sir is experienced pharmacovigilance professional for the last about 20 years plus and has worked in various capacities, starting uh, from clinical trials in his early days to case processing to aggregate report writing, signal detection, risk management, uh, developing the systems for pharmacovigilance departments, running them, managing them, facing the audits, inspections, conducting audits. So he's a wide range of experience in pharmacovigilance domain, and he has got good expertise. So uh, first of all, as a project, we would like to invite Gaurav sir for his session today. But before that, would like to quick sort of a voting. Perhaps is there a way you can raise hands is to or comment people who are already working in pharmacovigilance domain. So how many people are already in pharmacovigilance domain? So you can raise hand perhaps from the reaction side. If you see in the Zoom reaction, you can raise your hands, the people who are already working in pharmacovigilance domain. So we would like to do the quick vote so that we can, tweak the program accordingly. And the objective is this webinar was for the people who are not freshers, who have not worked earlier in pharmacovigilance. For them, we have a separate webinar next week. But this webinar, the objective is for people who are already in pharmacovigilance domain. Can you all raise hands, please? Can you all raise hands, please? And can we see how many people are already working in pharmacovigilance? Okay, so we have, so is that all people who are, we can see about 12 to 15 hands, already people who are in both pharmacovigilance domain, and the other people are not in pharmacovigilance domain.
Actually, there are people because I can see few of our alumni as well.
Okay, so you can raise hands if you're already working. That would help us in, modifying the session accordingly. And we may, for the people who have zero experience in pharmacovigilance, we can then talk very briefly, of course, in very detail next week. But I can see about 14, 13, 14 hands only, if that is correct. All right. So, Gaurav sir, we do have people who have no experience in pharmacovigilance domain. So, we will very briefly. I will not take Gaurav sir's time on doing that. Uh, the pharmacovigilance people who are new, who have no experience in pharmacovigilance, I'll spend about 5 minutes. It's a science of drug safety. Every pharmaceutical company needs to ensure that the long term safety of the drug from the point of clinical trials onwards till post marketing and after post-marketing, every company has to ensure the long-term safety of the drug. Apart from efficacy, the safety is most important, which requires the companies to, as per the various regulations and the pharmacovigilance guidelines, have to carry out the post-marketing activities, or even during clinical trials, the pharmacovigilance activities, which means all the adverse events, serious adverse events are reported properly to the regulatory agencies. You use the case processing for that. You do the aggregate report writing. You do the signal detection and risk management. All these things we are going to cover in much more detail next week in our webinar for the freshers. So today, the focus will be for the people who are already working in this domain, what they could do with this program in the advanced pharmacovigilance. Over to you, Gaurav sir. You can perhaps touch upon the PV a little bit for about few minutes.
Sure. Thank you so much, Kamal sir. And I welcome all of you to this webinar on pharmacovigilance and the advanced pharmacovigilance activities which are done as part of pharmacovigilance. I can see as many of you are already working in the domain, you would have some basic understanding of pharmacovigilance. So let's start with the session and try to project my screen. Just a moment. Screen. Okay. So I hope you're able to see the screen right now.
Yes, sir.
Yes, sir. It's visible.
Thank you so much. So again, the first thing that we all need to understand with pharmacovigilance is that we are required to keep the patient safety first. Whenever we talk of pharmacovigilance, pharmacovigilance is not a very old science, as we all know. It has come up recent, and if we talk of the thalidomide tragedy, that was the trigger point from where all the authorities started to get the awareness about having a pharmacovigilance system in place. Yes. So when we talk of pharmacovigilance, the safety of the patients is the first and the priority for any company, for any healthcare professional who works in the domain, right? Okay. So again, if you would have read this cartoon character, there was a cartoon character, Charlie Brown. The snippet used to come in the newspapers. Again, his friend is talking to him and saying, I think you're afraid to be okay, Charlie Brown. Don't you think medicines would be good for you? So he's wondering and saying, I don't know. What are its side effects? So that is the concern for everyone, that what are the side effects? What do you expect from the medicines? So 5% of all the hospital admissions are because of these side effects, and in Europe almost 2 million deaths have happened every year because of the side effects, what we call in common parlance as side effects. In again, in medicine, in pharmacovigilance, we call it as adverse events or adverse reactions. Yes, because those of you who are working would know the difference between adverse reaction and an event. We'll quickly look at that as well. Okay, for those who are new. Okay, let me proceed ahead. So there's this famous quote from the Dr. Vladimir Lefekin in the Geneva conference, which was organized by World Health Organization, saying that dying from a disease is sometimes unavoidable, but dying because of an adverse drug reaction is unacceptable. And I'm sure that those of you who are working will understand the priorities that the regulations put on the fatal and life-threatening events is only because of we don't want drugs to kill someone. Yes, we don't want medicine to result in death. Yes, And so this is one of the reasons why we need to have pharmacovigilance in place. Right? So during the COVID times, I'm sure that all of you remember that people were afraid of getting vaccination done. They were running away. They were not willing to get the vaccination done only because of apprehensions of death, of it resulting in death. Yes. So this is something that was worrying for people, because vaccines, we need to make sure that they are safe for use, because ultimately these are prophylactic. They are not there to treat the disease. They are there to prevent the disease. And so if somebody is dying because of that, it isn't acceptable. Yes. Okay. So as per the World Health Organization, we know that the definition of pharmacovigilance is that it's a science and activities relating to detection, assessment, understanding, and prevention. So everyone would have read this definition. Those of you who have been working in the domain would have read it. But then why do we call it a science? Is because it is evidence-based, is because we try to collect evidence of an event and to classify an event as an identified risk. That means we have enough numbers to say, yes, this is the event which is being caused by the drug, and we know the mechanism behind it. Yes. So that is why we call it a science. And the activity is that we are trying to collect the ideas, we evaluate them, assess them medically, understand them, why they are happening, and then we try to prevent them. So I always keep this example as a simile when you are faced in a real life situation. Suppose you are driving on a highway and you find that there is a speed breaker ahead. Yes, and you see a signboard on the corner which says there is a speed breaker ahead. Then what do? You would slow down. Yes, you would slow down and then proceed. So you will not get the jerk. Yes. So the same is the case with a medicine. Unless we know the signals, unless we know the issues which can come up with a medication, we'll not be able to handle it properly. Yes. You can manage a risk well if you know it. Yes. Isn't it? So that is what is the concept which is followed in pharmacovigilance in medicine, whether related to vaccines or again our medicinal products. The concept is the same, that you need to understand what is going wrong and then prevent it from happening. Okay. So again, like we were discussing, what is the difference between an adverse event? When I say an adverse event, it could be anything that goes wrong after I have taken a product, after I have taken a pharmaceutical product, which is given at any dose, but it may or may not have a relation with the drug. So for example, let's take an example. A patient took a medicine and went out of the facility or maybe the hospital, and he met with a road accident. Do you think it's an expected event? Do you think it was something which was expected to happen? No. So it's an untoward medical occurrence. Yes, because the patient has met with a road accident. Yes. Now it may be related to the drug or it may not be related to the drug, but it is an untoward medical occurrence. So we call it a adverse event. Yes. Is this point clear to everyone? Yeah. And when we talk of a reaction, so what is the difference between an event and a reaction? Now, in a reaction, it's a noxious or an unintended response which is given for a drug which is given at a normal dose in humans, either for treatment or for prophylaxis or for diagnosis of a disease, or maybe modification of physiological function. Yes. Okay. So that means it should have a relationship with the drug. So I gave you an example of a patient moving out of the facility and meeting with a road accident. Now, suppose if the drug was, say, anti-allergic drug, maybe a drug like, say, chloramphenicol, or it could be it could be any medication like cetirizine, very commonly used. Yes, and maybe it would have caused some kind of sedation, isn't it? The drug would have caused some kind of sedation. Yeah. Okay. And when we gave the drug, the event appeared, and when we stopped, maybe after a point, we saw that the patient was okay. Yes. So we can have a temporal relationship. We can attribute and say, yes, the drug would have caused the event. Yes or no? Right? So in that case, where we have a relationship established because we can explain it via a mechanism of the drug, or we have evidence to prove, yes, it is because of the drug. There are no other confounding factors. There are no underlying disease condition which the patient had except for taking the medicine, right? So in that case, we can establish the relationship and we call it a reaction, right? Okay, I'm sure this point is clear to everyone. So the difference between an event and a reaction is, again, in the reaction, you may or may not have, in the event, you may or may not have a relationship with the drug, whereas with a reaction, you have a causal relationship established. Okay. Again, for a valid report, what is the minimum safety information that we need to have? Yes. So we need to have, again, identifiable patient. I'm sure those of you who have worked on case processing will know. Again, I'm giving this for the information of others who are new to the domain. So an identifiable patient could be any person, either his name. So if I am Gaurav Grover, my name is my identity. Yes. Or it could be my initials. So GG. Yes. Again, I'm a male. So the identity could be a gender of the person. Yes. It could be, again, the age. That means, so a 40-year-old. Yes. Could be an age, or could be the date of birth of that person. Yes. So the date of birth could be an identification of a person. Similarly, his weight, his height at that point of time. These can also be his identity. Yes. Also, it could be the age group. So I may not know the exact age, but if I say an adult, yes, or an old patient, or a child, so that means I am giving a categorization to the identity. Yes. So that becomes a identifiable patient for me. Is that clear to everyone? Yes. Again, a contactable reporter. So we need to have a person who will report the case. Now, reports can come to us from anywhere. It can come from a registered doctor, a pharmacist, a nurse, yes, or even the patient can report. So again, we call it as a consumer. A patient may report directly, or maybe his relative. Yes. So if it is a child, maybe the father or the mother would report the case. Yes. Or could be a lawyer of that patient if he is, approaching a lawyer for any reaction. So it could be the lawyer of the patient. Yes. So they will be your contactable reporter. And how can we contact them? So you may have an email ID. You may have, again, you can have the postal address. You can have the telephone number. You can have the fax number. So these are the ways by which we can approach, yes, a person, right? So contactable reporter would mean that you have some way of communicating, right? Okay. Then we have a suspect product. Yes. Okay. So a suspect product, just a moment, give me a second. Okay. So like I said, again, a suspect product could be there and an adverse event. So these are some of the things that we consider as a valid report. Now let's quickly look at the pharmacovigilance system, when we talk of the pharmacovigilance system, you have the safety database, which we all understand that there are different databases which are available, and the standard operating procedures, which will govern how we process the case, how we identify a case as valid, how we establish the causality with the drug, right? And then we have a network of local or regional experts who will look at who are trained, who are experienced professionals, who will look at the pharmacovigilance system and say, yes, okay, this drug is related or not related, and what are the benefits of the drug, what are the risks associated, right? So this is what we will look at in the pharmacovigilance system. Okay, now how does pharmacovigilance system help? So as we all know, again, this is one of these characters, Snoopy, who's talking to his friend, and the friend says, Peanut says that, here the pill the doctor told me to take. So he says, I'll be interested to see if it has any side effects. Then Snoopy is thinking and saying, me too. One of the secrets of life is to have good side effects. So the purpose again of having medical review, of having assessment of cases, is that what we are trying to establish is what are the benefits of the drug versus what are the risks. Yes, what are the good side effects? That is what we are trying to do with pharmacovigilance, right? So as we've discussed the definition of adverse event and the reaction, the balance between the effectiveness or the benefit of the medicine versus the risk or the harm is what we are taking into account when we say, yes, the medicine is good for use in the routine clinical practice. Yes. So the comparative information has to be there between different therapies. How do we say, okay, when we say azithral is better than amoxicillin in respiratory tract infections, is because of the effectiveness of the drug, as well as what we say is, again, yes, the compliance is better because it's a once daily dose as compared to, again, amoxicillin, which might be required for more than once. Yes, and you need to follow a longer course, right? Okay, so comparative information is something that, and again, the adverse reactions that are associated with the medicine. Yes, so that will help us to decide what is the benefit of the therapy versus the risk. Okay, is that clear? Again, it's more useful when we are doing it for the pre-marketing information, because at that time, the drug is a new drug, it is limited, it has selected subjects who have been exposed. Yes, once we start to market, you have more information, and then you can establish the relationship. We will come across these concepts of pharmacovigilance, the risks that we call when we are establishing a benefit versus the risk. So the absolute risk is, again, how many people have been exposed to a drug and what is the probability of an event which will affect the people or the members of that population. So we call it a absolute risk. As an incidence, that means when it is measured over a period of time, we call it as incidence. When it is again done at a particular point of time or a given time, we call it as a prevalence. Yes, okay. Again, the concept of relative risk is used where we do the ratio of the exposed population. So the absolute risk, that means how many have been exposed and have had a reaction, and the risk in the unexposed population. So why do we say the COVID-19 vaccination was effective? Was because the relative risk is, again, where the drug causing the event was comparatively lesser. Yes, when the difference or when the ratio between the absolute and the reference population, which is not treated, if that shows a relative risk, if you see here, we've given an example that, okay, suppose rash has happened in a population after taking a medicine. Okay, so now say treated population was 1500 patients, of which 35 had reaction, which is rash. So then this is the ratio that we get for absolute risk, and then the ratio of this with those who have not been treated and have had a rash. So that will give us the relative risk, right? So the more the value, that means this 9.3 is the value. So the more the value, that means the more is the chance that the risk is related to the drug, right? Okay. So this is what we use, again, the relative risk, the absolute risk, reference risks, while establishing the risks associated with a drug. Okay. So benefit and the risk benefit analysis is done for any individual or for a population. We also call it as effectiveness risk or the benefit risk assessment. Again, the favorable outcome that we want. Okay, so if a drug is useful in treating a particular condition, right, and the undesirable effects or the unfavorable effects that we notice, so the ratio always has to be towards the benefits. The benefits have to outweigh the risks always. Only then we will go ahead with the treatment, otherwise we will not. Yes. Okay. So again, the concept when we are reporting a ADR, and I'm sure those who have worked on spontaneous reporting will know, listed and unlisted events. That means what is given in your label, in your prescribing information, in your summary of product characteristic, is what we are required to report to the authority. So SUSARs, or the suspected unexpected serious adverse reactions, are required to be reported. And so as a medical reviewer, your first job is to look at the listedness of the event. That means in terms of the nature or the severity or the frequency, whether the event is in any way related or not related. Yes, or is listed or not listed, is what we are looking at as a medical reviewer. Yes, so we are seeing whether the event is given in the label or not. Yes, in the SPC in Europe or the prescribing information in US, right? When we talk in terms of clinical trial, so the investigator brochure is the document or the developmental core data sheet. Yes, so these are the documents that we will look at, where we will see whether the event is listed or not listed. Yes. Okay. Again, like I said, the nature or the severity. So maybe earlier the event was mild in nature, it was causing rashes. Yes, but now when I am receiving it, I am getting urticaria or anaphylaxis or an anaphylactic shock, any severe form, or even a Stevens-Johnson syndrome, which is life-threatening, or say toxic epidermal necrolysis, which is again life-threatening. So all these events where the change in the nature is observed from what has been observed earlier. So IB or the investigator brochure is always updated with the new information, or even the developmental core data sheet. Yes, so we will always look at the event and see if that is given in the label or not. If not, if the report is not listed, it is not there in the label, then it means that it is a unexpected event. Yes, is that clear? Okay, so as we were discussing again on the medical review part of the case, as a medical reviewer, you need to look at the listedness or the expectedness of the event, and then we will look at whether the drug, once we stop the drug, if the event stops, we call it as a positive D challenge. Yes, so whether the event is in any way caused because of the event, because of the drug or not, we will establish that and see whether the event is related or not related. So for that, we need to have the D challenge and the re-challenge information. So in the D challenge information, the positive D challenge, that means, whether the stopping of the drug, the event completely disappears or partially disappears after the withdrawal. So we call it as the positive D challenge. And if it still continues even after stopping of the drug, so we call it a negative D challenge. Yes. And then we also apply the concept of re-challenge, where when we again reintroduce the product and the event occurs in a similar signs or symptoms after reintroduction, then we call it as a positive re-challenge. If it doesn't, if there is no signs or symptoms which are similar to what was observed earlier, we call it a negative re-challenge. So these are the things that we look at when we are establishing the causality of the drug and establishing whether the drug has caused the event or not, right? So again, we look at the seriousness criteria of the event. As a medical reviewer, we need to look at whether the case is serious or not. If it has resulted in death or is life-threatening, requires hospitalization, results in disability of any kind, persisting disability, any congenital anomaly, and if it is having the medically important condition, it's a medically significant change in nature, frequency, severity. So we will call it a serious adverse event, right? Okay. And I'm sure you are aware of the spontaneous and solicited report concept. So which is that in a clinical trial, whatever we get is a solicited report. Whatever comes from marketed reports is the spontaneous report. So what do we do in the causality assessment? Again, the evaluation is done of the likelihood that the medicine has caused the event. Again, causality assessment is usually done with various algorithms. Yes, so we use different algorithms to do the causality assessment. The most commonly used algorithm is the WHO criteria or the WHO UPSLA monitoring center scale, where we are categorizing the events as likely, certain, probable, possible, doubtful, not related, or unaccessible. Yes, again, the different causality scales which are used. So we have, apart from the most widely used WHO scale, the WHO UMC scale, you also have Naranjo scale. But the most problematic issue with Naranjo scale again is that in Naranjo scale, the least that you can go to at times is possibly related, because the very first question in the Naranjo scale says that, did the event appear after the drug was taken? If you say yes, the rating goes to +1, right? And so that means anything above 1 is possibly related. So the least that we can go to in Naranjo scale is again possibly related. But then there are several other advantages of using Naranjo scale. Again, that Naranjo scale allows you to give a consistent result every time. There are no variations between the medical reviewers. So medical discretion can differ between the two people. But then with Naranjo scale, you will have a uniform result, right? So this is again something that with the causality assessment, using Naranjo scale, there are advantages that, and that is why many clinical trials we use Naranjo scale because of the uniformity. However, when it comes to widely being used, so again, WHO UMC scale is more widely used because, like I said, here you apply the clinical judgment, you apply the discretion, you look at the temporal relationship, you look at the D-challenge and the re-challenge, and you are applying whether the mechanism of action allows this kind of a reaction. So we are putting our own clinical judgment in the WHO UMC scale as opposed to Naranjo scale, which is totally a scale based. And why there is a particular rating given in the Naranjo scale is also not very clear. So the weightage is not clear in Naranjo scale because we don't know how and why a particular question has been given more rating or less rating. Okay, let me stop to share the screen for a second. I will share the Naranjo scale with you now. Just a moment. Okay, just 1 second. I'm trying to share my screen. Okay, so as I was showing you, this is the Naranjo scale where you have a probability scoring. There are these standard set of questions which you need to answer. Again, in these questions, are there previous conclusive reports on the reaction? Yes or no, or don't know. Now, why the rating of +1 has been given or why a rating of -1 has been given is not established anywhere in Naranjo. So this is the problem. Again, this scale was developed by Naranjo, who's a researcher. And the problem, like I said, with this scaling is why for a particular question, there is a +2 or a -1 given or +1 and 0 for another question. So this explanation is not there anywhere. This is the problem with this scale. However, like I said, the benefits with the scale is that, again, the scoring that you get in this scale will be consistent because you have to give any of these three only as a reviewer. So if you had a previous conclusive reports of a reaction, you can say yes or no, or you don't know, and that will give the overall score. Yes. Okay. So there can be nothing other than these three. So the consistency is there. However, like I said, the problem is one, that why this kind of rating has been given, and then it is also biased towards the reactions. If there was, reports which were reported earlier, so for them it is biased. If there are conclusive reports, it is giving yes as rating of 1. If there were no previous reports, that means it's a new event. So for that, it doesn't give any rating. It gives a 0 score. Yes. So it is biased towards previous knowledge of an event also. Okay. Is this point clear? Yeah. Again, with the scoring, if you will see at the scoring overall, so if the score goes above 1, like I said, so the least that you can go is possibly related, because the second question says that did the event appear after the drug was administered, which will always be yes or no. The event will happen after the drug was given. So that means yes will always come. So the least that you can go with Narengio is possible because it is falling in the range of 1 to 4. Right? Okay. So this is the thing with Narengio scale. Again, the rating, why it has been given +1 or -1 or -2 is not clear. Right? Okay. Now let me stop to share the screen. Okay. And I will go back to okay. So before we proceed again, like I was explaining, the WHO UMC system for causality assessment is more widely used. Again, because with the one case report, we can do the assessment using the Bradford Hill criteria for causality assessment, which was developed by Sir Austin Bradford in 1965. So once the reports are suspected, again we can individually assess them, summarize them, and review them for any additional evidence. And based on the strength in the case, so if we have enough information about the D-challenge, re-challenge, and the background information or other positive underlying disease condition, and all these factors can be taken into account when we are doing the Uppsala Monitoring Center causality assessment. So then we can identify, based on these, we can even identify a signal, right? So that's one of the reasons why UMC is more widely approved, because you can apply clinical judgment, right? Okay. And but the only discredit is that there is variation. There is variation in the WHO scale, because one person may have a different assessment. You may look at the pharmacological aspects or the case history or the quality of the documentation, and you will have a differing opinion, right? Okay. Okay. Now, let me move ahead. Let me go back to the slide deck. Yeah. Okay. So next again, based on these reports, whatever individual cases that we receive, there is a concept of aggregate reporting that we look at. Now, what is an aggregate report? Aggregate report looks at the cumulative safety information from various sources or wide range of sources, from the clinical trials, from the marketed reports, from the literature reports, which are published in the peer-reviewed journals, yes, or even in workshops. So whatever information sources we have, based on that, we will review the cases periodically. Okay. So in aggregate, as the name suggests, aggregate report comes from combined or combining all the reports that you receive in a defined time period. Okay. So on the periodic basis, we get the aggregate reports. And again, the findings which are there from the regulatory authorities worldwide are considered in the aggregate reports. Okay, so they play an important role when we are doing the assessment of the medicine, because we are looking at all the risks, all the new reports, all the ADRs which we have received in that time period versus the benefits. Okay, so that is why aggregate report is also called as a periodic safety update report, because it is done periodically. And now the new name for the periodic safety update report is the periodic benefit risk evaluation report, because we are reviewing the benefits versus the risks. Yes, so in the periodic benefit risk evaluation report, we are comparing all the reports from literature, from spontaneous cases, from clinical trial cases, and our signal detection activity. So all of them are combined, and then we make the assessment whether this signal is a new signal, or it is is it an identified signal, or is it a potential signal. Yes, and when I say identified, that means, do we have the evidence that yes, enough numbers are there, we understand the mechanism why this event is happening. So that is called as the identified risk. Yes, so identified risk is enough numbers of cases, and the mechanism, what is the mechanism for that event. So that becomes our identified risk. And the potential risk again could be the information is missing. We do not know the basis or the mechanism why the event has happened, how it has happened, right? So again, the classic example is the blood clot related issues with COVID-19 vaccine, Covishield, which was sold, or Vaxzevria. Again, this blood clot related issues, we still do not know the mechanism why it was happening, how it was happening. Okay, the risk factors are known, that yes, if there is an underlying disease, then the patient can develop blood clot related issues. But why is it happening, or we do not have the enough numbers. So that is why it is still a potential risk. Yes, and we need to collect more evidence for that. Okay, so in the aggregate report, we try to evaluate the cases from wide sources, from wide range of sources. Okay. So like I said, it is a compilation of all the newly available safety information during the defined reporting period, and the type of report will vary according to the country or the regulations. So in Europe, we are submitting the PVRER, yes, which is the periodic benefit risk evaluation report. They call it a periodic safety update report. Yes. So in the PSUR, what do we submit? We are submitting all the reports from all the regulatory actions which have been taken on the marketed products or on the clinical trial products with the same active ingredient. Yes, then we are also analyzing all the reports received from marketed products, all the reports received from clinical trial cases which are serious adverse events. Yes, and then all the literature cases, the reports which are published. and then we analyze all these newly available safety information to conclude if we need to do any risk management. If there is a new risk, maybe we will need to, are we able to manage it with the routine pharmacovigilance activities? So routine pharmacovigilance activities, as we know, are ICSRs, which you process and report, or the periodic reports that we will make, the aggregate reports, right? And the signal detection that we do. But do we need to do anything beyond the routine? So which is your benefit risk evaluation? Yes. So anything beyond the routine. For example, let's take a classic example of a drug like isotretinoin. We know, again, isotretinoin is used in acne. Yes. And the most common side effect, again, of this drug is that it is a known teratogen. It can cause birth defects. So then there needs to be some additional activities, just merely mentioning on the label that this drug should be used safely while giving to pregnant women will not be enough, or it is contraindicated will not be enough. You can't control if some pregnant woman takes the medicine and has some problem. So in that case, what do we do? In that case, there are some measures that you need to put in place, which are your additional pharmacovigilance activities. Yes, which is a part of your risk management planning, right? Okay, so that is what we look at in the aggregate. In the aggregate report, we will look at the new risks which are coming out even from the signals. Yes, and then accordingly, we will suggest an action that whether we can manage it by updating the label, or we need to also do some activities. So again, like I was telling you, blood clot related issues with COVID-19 vaccine, Vaxzevria. Again, these blood clot related issues have been updated in the new label. Yes, because it is a potential risk and there have been deaths because of the blood clot related issues. So you can't allow someone to die because of a medicine. Yes. So that is why the routine minimization measures were taken. That means the label was updated. However, additional have also been done now, and the authority has asked for some specific targeted studies, post-authorization safety studies, to look at the risk related to this particular concern, the blood clot related issues, right? So this is what we will look at in the aggregate report conclusion. At the end, we will suggest what we need to do, whether we need to monitor this effect more in future reports or we need to do a risk management, right? So why aggregate reporting is important? Like I said, you need to do detailed analysis and evaluation of the benefit risk ratio of the product. Again, it is not possible by doing only review of the individual cases. You can't do that. If you have one case or two cases, it will not be possible to see how much benefits are there and how many risks have been reported. Yes, but if we are doing it on a regular basis, in a periodic basis, then it is possible. Yes. So periodically reviewing the safety reports which are received either cumulatively also is significant. That means what in the PDRER now what we are doing is we are also looking at from the date of authorization anywhere in the world for the first time. All the reports are being reviewed. Yes, we are analyzing them and seeing how now whether the risks have changed, have the benefits been outweighed by the risks? If yes, do we need to take any special attention? So these reports need special diligence and attention to the detail. Also, what is essential on the other side, we need to also look at that. Okay, what is the minimum thing that we need to do, whether we can handle it by routine activity or we should suggest a additional risk minimization as part of the risk management planning. Okay, so this is what is done in the aggregate report. Again, the aggregate report is important to identify the safety information which will require further investigation, like I said, of the product. and the changes to your label. So what changes you are suggesting in the label? Yes. And it will also provide a concise summary of the safety information. It will play a key role in doing the assessment. So what are the new signals that you have got, whether you need to do any other additional activities. Again, the exact type of report will vary according to the country and the regulatory authority. So in US, the report which is there is the PADER, which is required instead of the PBRER. US has a different timeline. Again, instead of the PSUR, you are required to submit PADERs, which are quarterly submitted, yes, from the date of approval. Again, in US, the reports that we make are product specific. They are not active moiety based, unlike the PBRER or the periodic benefit risk evaluation report, which is made as per the active ingredient. Yes, but in US, what we are making is the product specific report. PADER is again periodic adverse drug experience report. Yes. Okay. So which is product specific, and it is shorter in duration as compared to a PBRER. So in US, like I said, from the date of approval, we are required to give quarterly PADERs for the first 2 years and annually after that. Whereas again in Europe, we are required to submit the PBRER, which is an active moiety based report, where all the formulations have to be included, all different types of reports. So if I am selling paracetamol, I have to take into account the injection formulation, the tablet, the capsule, whatever, suspension. All of that has to go into the PBRER and cumulative data. Whereas again in the PADERs, which is again the US specific format, you are only making a product, and that too one type of formulation in the product, and within the interval only, which is quarterly. But why do you think US is asking for quarterly PADERs, whereas Europe asks for PBRER? Yes. So the difference is that again the database, the safety database of US has always been the single database. Yes. So since 1950s onward, they've been using the same database in US. And so all the reports are going into that same database. Whereas in Europe, you have different databases. Earlier, every authority was having their own database. So MHR, and some were not even having in Europe. So MHRA from UK had their own database earlier. French agency, again, AFSAP had their own. Okay, German BfArM had their own, but there was no one single repository. But after the 2010 pharmacovigilance legislations, in July 2012, it was decided that the UTRA vigilance database will be set up. And the UTRA vigilance database now relies on this cumulative report, which we give in the PBRER, right? Okay. So this is what is required in aggregate reporting. Again, we have different components within the aggregate report. So you have the worldwide authorization, you have regulatory actions due to safety reason, which are coming into the report. The risk management plans are a part of the PBRER also. Yes, so we are looking at the risk management plans, the spontaneous reports, the clinical trial reports, the literature reports, any studies which are being done, safety findings which are coming from the non-clinical studies, patient support programs, and again the competent authorities. These are all a part of our sources in the PSUR or the PBRER. Okay. So like I said, you have different types of aggregate reports. There can be some reports which are for the marketed, and there can be some which are in the trial phase. So these aggregate reports which are made in the trial phase are called as the annual safety report in Europe or the IND annual report in US. Now these have been replaced by the ICH guidelines as the DSUR or the Development Safety Update Report. Yes. So this is what in the in the clinical trial phase or the pre-approval phase, these are the reports that we make, aggregate reports. Whereas when we compare it to marketed, like I said, in US you have the PADER, Europe you have the PBRER. There is also at the time of renewal in Europe, ADCO, which is Addendum to Clinical Overview. Yes, and then we have the risk management plan that is again made based on the various sources of information. Okay, now some of the important terms, I'm sure everyone knows about it. The international birth date, the date when the product was first authorized anywhere in the world. In the trial phase is called DIBD, developmental, right? Okay. Okay, let's move ahead to the DSUR. Again, like I said, DSUR is a comprehensive report which is done annually. So unlike the periodic reports or the PSURs, which are for marketed products, which are done 6 monthly in Europe for the first 2 years, annually for the next 2 years. Yes, and then you have a 3-yearly report. So that compared to the trial report, we have the DSUR, which is done annually. Okay. Again, earlier, what was happening in the DSUR and the PSUR, when we were making, so IND reports were only focusing on in Europe or in US, the reports, annual reports, which were being made for the clinical trial reports, they were only focusing on clinical trial data. Yes. And similarly, the PSUR, which was required in Europe earlier, was only focusing on the periodic data reports from marketed products. But now, both the PBRER and the DSUR are looking at all sources of information, because you can't look at information in silos. It cannot be that, clinical trial, whatever is there, this is only related to, clinical trial. I will not look at. It's like two friends who are angry with each other, and one says, I will not look into the other person's, notebook. Okay. So, but eventually the drug is the same, whether in trial or in marketed. Yes. So whatever information is coming will impact the overall safety of that drug. So that is why now in the DSUR, we are doing a comprehensive review. We are looking at all sources, whether it is from trial or it is from the marketed source. Okay. Again, this is driven by the ICH guidelines. The ICH E2F guidance covers the requirement for DSUR, which has been adopted by all the regulatory authorities, whether Europe, whether US, any part of the world. Right. Okay. Again, like I said, in the DSUR, you have all these terminologies of developmental international birth date and the data log point. Again, in clinical trials, we also have an end of study report where we are giving the summary of all the summary of the safety data which is received in the trial. So all the serious adverse events are collected and summarized in this report. Okay, right. Now, annual safety report, we have covered the concept already. And this is the submission schedule for the DSUR yearly, for PSUR, like I said, 6 monthly for first 2 years, annually after that, and then 3 yearly. Okay, PBRER, this is what now we have in place. Okay, PADERs, US, like I said, quarterly for the first 3 years and annually for the next 2 years. Okay, so this is what, and then the submission timelines are now 30 days and 60 days for quarterly and annual PADERs. Okay, again, let's move to the, just give me a moment. Okay, let's move to the signal detection. Again, when we talk of signal detection, now, what is a signal? Signal is something that stands out from the background. Again, in computers, this is a concept which is used. When we apply it to medicine, anything where we can associate our drug with the event, okay, is what we call as a signal. So this is most commonly used for post-marketed phase, but it can also be applied to the clinical trial phase. So signals are anything where we have a doubt that the drug may have caused the event. Again, the CIOMS working group says that it could be one or multiple sources from where you can get a potentially causal relationship, okay, or aspect of a known association. So it was known earlier, but now we are understanding it better between the event and the and the drug. Okay, so but where we have a likelihood that the drug has caused the event, it can be judged with a likelihood. Okay, so that is called as a signal as per CIOMS. Same way as WHO also has its own definition that there is a causal relationship between the event and the drug. Again, the relationship was not known earlier. It was not properly documented, but we need to have more than one report to call it a signal as per World Health Organization. In EMA, again, the safety signal is a new or a known adverse event where there is a potential, possibility of the medicine causing that event. Okay, so signals are generated from different sources. Again, they said you should refer to spontaneous, clinical trial, and scientific literature. Okay, so all these definitions are a bit different, but eventually whatever differences are there, still there is one point which is common in all of them, that potential harm that can be caused by the medicine and where you need to evaluate it more is a signal. Yes, so signal is nothing but it is always a hypothesis. It is a work of hypothesis. We are trying to estimate, we are trying to understand and suggest, okay, that this is a signal is based on our assessment of the existing data from clinical trials, from marketed reports, from literature reports. We try to work around and say, yes, the drug may have caused it, or the drug may not have caused it, based on the other conditions. Okay, so we need to either refute it by collecting more evidence as much as possible, or we will accept it, yes, based on the evidence. Okay, so signal is nothing but it is a relationship between the event and the drug. Yes. So like I said, now in the post-marketed reports, because you get so many reports, so that is why the assessment is always done by looking at the patterns or by looking at new findings from the safety database. Yes. Okay, so signals are again, like I said, from different sources we can get the signals. We can get it from unsolicited, which is your spontaneous reports, and from solicited reports, which is again you can get it from clinical trials, from registries, surveys, even from some research databases, survey done by the company, yes, or even named patient programs. So all these sources can be our sources for the signal. And the contractual agreement. So again, the most classic example, again, let's see, COVID-19 vaccine, Serum Institute getting into an agreement with AstraZeneca. Yes, so they will exchange the data between themselves. So the data from India will be exchanged with AstraZeneca. Yes, contractual agreement. Clear? And we can also get it from regulatory authorities, from the regulatory authority databases. So company also review the authority databases like US FDA's FAERS system, yes, Federal Adverse Event Reporting System, or for that matter, even the European Ultravigilance database. So they will all do their own, and the global database, which is Vigibase, maintained by WHO, UMC. So all of these are looked at for various sources. Any ICSRs coming from the authority will be reviewed for signals. Okay. Then there are different types of products which are available for data mining QScan by Drug Logic, Empirica by Oracle, Oracle Clinical. Oracle is giving this Empirica signal. AG signals by ARIS-G. Cerner has DS Explorer. So different, Empirica signal databases are available, which will help you in identifying based on various proportionality ratios like PRR proportionality ratio, or the reporting odds ratio, ROR ratio, which are looking at how you classify based on the statistical tools. And again, those signals can further be confirmed by clinical evaluation of the cases, right? Okay, so signal databases which are used again in marketed products. You have different types of signal databases, like we have the spontaneous reporting systems, VGBase, as I told you, or Vigilance, or AERS in US. Similarly, UK has Sentinel database. Okay, so there are several advantages, that it is highly sensitive, it can detect rare events, and it is inexpensive because these are maintained by the authorities. Yes, but there are disadvantages, like it requires reporter to identify biased reporting, under reporting also, like in US, maybe some events will go unnoticed, will not be reported. Yes, or there can be overstimulated reporting. So during the COVID period, you would have seen everyone was reporting in India, fungal disease. Yes. Okay. So these are the things which can be noted with signals. Again, the prescription event monitoring, or various databases which are looking at the prescriptions. Yes. There are problems like you have small size in those, and these are more expensive because you will have to buy the access to them. Okay. Then there are large link databases, healthcare databases. Okay. Which are again sponsored many a times by the government also. Okay, these are large, they are having large population, but you don't have the complete medical information in them. And then there can be electronic medical records like the General Practice Research Database in UK. Okay, now, so signal generation can be done through various sources, like I said, from preclinical, clinical, and we are looking at the pattern or the cluster of these events. Once you have identified the signals, and the signals are detected, you apply the statistical tools or the methods to calculate the association of those signals, and then those signals will be further analyzed. You will refute them by analyzing the cases individually of those signals. Yes, and if they are inconclusive, then the authority can ask for post-marketing studies or observational studies. Okay, right? Okay, like I said, the most common methods that you will use are PRR ratio or RR ratio, ROR ratio, reporting odds ratio. There is a Bayesian confidence BCPNN method which is used for WHO monitoring, and there is a multi-item gamma poison shrinker model which is used by US FDA. Okay, for MHRA. Okay, now once you have the signals, okay, so then you have to do the benefit risk assessment. Now, when it comes to benefits, what are the benefits? These are the positive outcomes or the favorable effects that you see with the drug. Yes, and benefits may be to a class of drug, or drug having a greater efficacy, being able to treat a broader range of patients, or being easy for compliance. Like I told you, azithral being a very common example, azithromycin, patient compliant for respiratory tract is effective against broad range of gram negative, gram positive. So you will be able to treat more patients. Yes, and there are the benefits associated with that drug, right? Again, the risks would be the undesirable or the harmful effects related to the drug therapy. They may be documented or suspected. They are again common or rare, severe, or it might be irritating, or you may be able to prevent them, right? So these are the things that we will look at the benefit risk assessment. Again, in the risk management in pharmacovigilance, what we will look at, I told you, you need to look at the number of cases to identify a risk, Okay, so if you have enough number of cases, you have the adequate evidence, yes, and you know the mechanism, so you will call it a identified risk. However, if you have some basis of suspicion, right, but you don't know, you are not able to confirm whether it is related or not related, or there are some other underlying conditions, so then we categorize it into the potential risk category. Okay, and accordingly we will do the risk management. Now risk management again, for the important risks. Now what are important risks which will have the implication on the public health overall? Okay, so in risk management planning or pharmacovigilance planning, as it is called in ICH, okay, what we do is all the identified risks or the potential risks that can have an impact on the benefit risk balance, these are categorized as important identified and important potential risks. Okay, and you need to take actions or you need to create activities to minimize these risks. Okay, is that clear? Okay, so the goal is again to figure out whether the risks or the benefits, what is more important. Okay, so it is often used in medicine because every medicine has some risks with it. No drug per se in totality is safe. When we talk of paracetamol, even paracetamol is not safe, yes, because you have even a rarer condition like Stevens-Johnson syndrome also reported in few cases, yes. So we have to look at the beneficial effects and also statistically how much harm can be done than the good. So we have to always weigh and see that the risks are outweighed by the benefits. Okay. So this is what we do in the benefit risk analysis. In Europe, we have the risk management plan, which is done for every product which is marketed. In Europe, it is a requirement as per the pharmacovigilance legislation that we need to make a risk management plan for every product. Yes. Whereas again in US, there is a REMS, which is the risk evaluation and mitigation strategy. Yes, which is only for certain products that have been identified by again the US FDA. So US FDA has taken the accountability to decide which products should have a REMS, whereas in Europe, the company has to evaluate every product and then say whether a risk management is required or not required for all the products which are authorized. So whether it is a generic product or a branded product, it doesn't matter. If there is a risk associated, you need to have a risk management plan in Europe, right? Okay. Whereas in US, only specific products where US database has identified signals, okay, where they feel there is a significant risk with certain products. So for them, they have asked for the risk evaluation and mitigation strategy. Okay, so in benefit risk assessment, what we are looking at what are the risks, how likely are they going to happen, what are the benefits, and how likely they will happen. Okay, so this is what we are looking at, the benefit risk assessment in medicine. It is a continuous process of evaluation, and we are looking at favorable versus the unfavorable effects. Okay, so it has to always outweigh the risks. The benefits have to outweigh the risks always. Okay, right? So risk management plan describes the pharmacovigilance system. It tells about all the identified, the potential risks, and the missing information. It states whether the routine PV activities are enough or we need additional activities. Like I said in isotretinoin, you may need to also do in regular routine pregnancy negative tests. Yes, to ensure that isotretinoin doesn't cause birth defects in children. And we need to do registry of patients. We need to register the doctors. We need to register the pharmacists. We need to give educational material to the patients, to the doctors. Yes. Okay. So these risks may need additional activities. So in the risk management plan, we will define those based on the risk profile of the drug. So for every important identified and potential risk, the risk management plan will tell whether the company is proposing any routine risk minimization, like I said, updating the product label, or you need educational programs, you need patient support, you need registries, you need tests being done at the centers. Okay, so those are the additional activities that you need to do in risk management plan. So it is a document that gives you knowledge about the current product, okay, the safety and the efficacy, right? And RMP provides the key information about what studies you need to do further to gain more knowledge about the safety or the efficacy of that drug. Okay, so post-authorization safety studies or post-authorization efficacy studies. Okay, so benefit risk assessment will involve looking at quality and the quantity of the benefit, the quality, quantitative and qualitative analysis of risk. So how many risks are there? Okay, again, we are comparing them to the natural history of the disease. So say for COVID-19, what was the risk? Okay, many people were dying untreated. Yes. Okay, so that is the risk associated with the natural history of the disease. It became a pandemic, and so we had to introduce emergency medicine. Yes, the vaccination. Okay, so benefits and risks of the therapeutic alternatives. Do we have any alternatives? Are there other treatment options available? Yes. Again, so there were not many options for COVID-19, right? So this is what we need to look at. Again, the ratio of the benefit versus the risk. This is all which is looked at, and we arrive at, again, different approaches are taken while doing the benefit risk assessment. So there is a multi-criteria decision analysis, MCDA, and decision tree modeling, which is done. Again, these are statistical tools which are used while doing the benefit risk assessment. Okay, and so large
Gaurav sir,
databases.
last few minutes we need to wrap up. Gaurav sir,
Yes, we are wrapping up. We are at
yes,
the
sir.
end of the session, sir. Okay, so factors that determine the benefit of the risk, like I said, you need to actually look at what is expected or what are the benefits for a particular individual. We also need to look at the evidence to support the risks. So how much uncertainty is existing for these risks? Are they serious in nature? Is it causing sequelae? How often is it going to happen? Okay, so blood clot related issues you would have heard resulted in stopping of the COVID-19 vaccine of AstraZeneca initially in Germany and Austria, Belgium. Yes, many of them had stopped. But then again, when they compared the benefits versus the risks, so very few deaths had been reported. And even in those cases, there was an earlier existing cardiovascular issue in those patients. There was an alternative cause. Yes. So that is why the benefits were outweighing the risks. And so they allowed to have this, product continued. EMA added the risk. That means the blood clot related issues can happen in those who have cardiovascular issues, right? That was added in the label, and they allowed the vaccine again. Yes, in Europe. So what are the risks of the disease or the condition if it is not treated? So if we would have left COVID-19 untreated, millions would have died. Yes. Okay. So the benefit risk balance is always done during the marketing authorization. The regulators will need to assess these benefit risks, and then they will arrive at a decision whether the profile is in positive or in negative. That means the risks are more or the benefits are more. Okay. So once this is, so always the benefit risk will keep changing. It is only valid at a point of time. It may change later on. It doesn't remain fixed. So new information on the benefits can come up later. Yes. So like in COVID-19 vaccine, India, they had said those who have been vaccinated have a better survival rate than those who have not been vaccinated. Yes. There was a study done in AIMS. So like this, new information on benefits may emerge once the drug is on the market and is being used in the clinical practice. Because again, you have more number of patients than in the trial. You can look at the rarer events. It also gives you the real world scenario. Yes. So there can be other diseases. In trials, you exclude patients who have pre-existing disease. But in real situation, when COVID-19 vaccine was introduced, patient had diabetes, they had hypertension, and they were taking the vaccine. Yes. Okay. So post-marketing studies may be ongoing to see the benefits in other indications, like they were testing it in children still. Yes, it has been approved in adults. Okay. So benefit risk balance is regularly done in the pre-marketing, and we are doing it after marketing, 6 monthly in Europe. Every 6 months, this benefit risk assessment is being done. And then we are evaluating cumulatively every time. What are the risks from the date of first authorization? Okay.
We'll take the questions now.
Okay.
Thanks, Gaurav sir. Thanks a lot. Thank you very much for your session. Before we take the questions, I'll ask my colleague to display the program brochure. We have an advanced pharmacovigilance program for the professionals who are already in pharmacovigilance domain. This program covers, starts with the medical review for the both post-marketing and the clinical trial cases. And then we fairly fast move to the aggregate report writing, which covers all 4 reports. We covered the PSUR, we covered PABRER, PADER, DSUR, and other aggregate report formats. And a lot of hands-on training in this program where the experts like Dr. Shiva and Gaurav sir will be taking you through the program. And, finally, you'll be writing the report. PABRER is the one example where you'll be writing a complete report. Then there is extensive training on signal detection, risk management, and also in signal detection, we use the, signal databases. After that, there is after signal risk management, we also cover all essential documents like pharmacovigilance quality management systems, audits, inspections, also things like safety data exchange agreements, pharmacovigilance system master file. So this is the extensive training program for people who are already in PV, in case processing part, or have some medical review experience. For them, this program is fairly useful. The program is commencing end of this month, the last Sunday of July. And this will be a 6 months duration program with the classes on Saturdays and Sundays. In this advanced, postgraduate certification program, we cover all aggregate, signal detection, risk management, medical review, and all essential documents are well covered, plus audits and inspection preparedness. So the fairly comprehensive 6 months program. Shortly, we are going to share the brochure and all information with all the participants. And now we are open for the questions. We are happy to take. You can unmute your mic and ask the question.
Everyone can now unmute
and ask one by one. You can raise hand and ask the question so that
you'll
we
be
can
allowed
answer
to
everyone.
unmute now.
Yeah. You can ask questions, please, in case you want to ask questions.
We do have. Yeah.
Good afternoon. I just wanted to ask, what is the course that you offer for beginners who have no experience in pharmacovigilance and they have, but they have clinical experience?
Clinical means, sir, you're a medical doctor. Please tell me your qualification simultaneously. If you introduce yourself, tell your qualification.
Yeah, I am a general physician. I'm a doctor with around 14 years of experience.
14 years. 14 years is the MBBS.
Yes, post MBBS.
Okay. Okay, Pusha. All right, sir. So there is a course, the same program, Advanced Drug Safety Management Program, which is for the physicians, called the Drug Safety Physicians Program. So it's taught simultaneously, and that is for the doctors, Drug Safety Physicians Program, where if you do not have any sort of experience, you will get the same program but a different certification as Advanced Drug Safety Physicians Program, designed specifically for the physician. So that starts simultaneously end of July, on Sunday, last Sunday
Yeah.
of July.
So after finishing that program, is it possible for me to do remote work in any of the
Sir,
pharmacovigilance?
remote work, no work, that is not the criteria we offer these programs. Remote work or the office-based work is the between you and the employer. We have no role to play on that part. If the employer is willing to offer you a remote role, that's their call, there's their policy. Clinomines has no role to play. Our job is to give you the solid training, prepare you for that role, and also in the process for the freshers, who have no experience in PV, also arrange the interviews with the recruiters, but not in the advanced program. For the people who are already working in the industry, for them, it's an advanced skill role, but the placement support is provided to all the freshers who are first timers in the industry.
All right.
But
all
whether
right.
it's a
Yeah,
remote
and
or
can I
office-based,
add
that's
to what?
between.
Sorry, sir. I can just add to what Kamal sir was saying, that most of the employers now are going back to full-time working or hybrid mode. Online is something that employers are now moving back to the office location. So that is between the employer and the employee. There is nothing that Clinomines will do as such. But yes, this is now the norm that they are going back to the normal mode.
And sir, and all to the freshers, at least all people who are working here, a different story. But for all freshers, in case you really want to get into the industry, be prepared to work from office, because all the fresher hirings are happening now from office only.
Okay. Thank you.
Yeah. Any more questions, please?
I'm Dr. Rajlakshmi. I finished my MDS in oral maxillofacial surgery.
Sorry, ma'am, your MD in?
Yeah, MDS in oral and maxillofacial surgery.
MDS, okay.
Yeah, MDS. So, and I worked with ICSRs for 11 months during the COVID period. I have 8 years experience in clinical practice. So, which course do you suggest that I would take at this level? And
what did you say your current experience in IQVIA is?
11 months. I just worked during the COVID period because it was offering remote location.
What job role?
PV basic associate safety.
Associate safety. So if you have as 11 months experience, I would say as good as that. We have a different program for you, because there you are going to learn in a more structured way the starting part of the pharmacovigilance, all regulations, essential documents, case processing, because 11-month experience is as good as probably from a recruitment point of view. After the gap, especially, I think go back to the basic role of a drug safety associate, either without doing course or after doing the course. But go back first, learn the drug safety associate or the case processing role a lot more, acquire 2-3 years experience before you want to go into the advanced pharmacovigilance domain, for which we have a different course actually.
Okay, so do you have brochures for that also?
Yes, ma'am. There is a course in pharmacovigilance. It is a separate program, and my colleagues Vinayak and Rajesh would reach out to you after the session. Then.
Thank you so much. Thank you so much, and it's a wonderful session. Thank you for that.
Thank you. Thanks a lot. What is the cost? So there's no cost. This is the fee, Dr. Anupam Mahajan. There is a course. This course is 59,000 rupees plus 18% GST is the total 6 months program for this particular program. And we will send you the information.
Thank you.
Connect with us also on the WhatsApp.
Hi, good afternoon.
Good afternoon.
I'm Preetham. I have around about a little approaching 10 years of experience in PV.
Okay, sir.
So I have certain basic questions. I have done a course from Cliniminds in 2018, which is aggregate reporting only.
Okay.
So I'm just giving you the basic understanding. I found your course is a little bit costlier than the market. So do you have any other sources? Because I was very interested in business intelligence course which you have, but it is little in the higher side, so I could not enroll it. So do you
So
have?
fees is something we cannot, discuss higher or lower. That's something for you to decide. In case you find the value, please do it. Over 1,000 students every year do. They find value in the program. I do not wish to get into that discussion here. You can talk to me one-to-one. Fees is higher or lower. That depends upon the quality, delivery. And in case you're asking me that where else you can, that's your research. Mr. Preetham, you can go and find out, and where else you will get in a more structured way the proper teaching and learning mechanism. The programs are always taught in a more structured way, and of course there is a Google search will lead you to a lot of free information on internet, but free is always free. When we pay for anything, and here we have a track record now, not 1 to 5 years, almost touching 20 years now, of constantly delivering the quality training program. 20 years is not a short time, Preetham sir. And we have been, constantly running these programs, offering to the industry experts. We run the corporate training programs for the corporate clients. Every pretty much a large CRO or a pharma company, we run the training program. Now, in terms of your business analytics program, please connect with us on one basis. This is not the right forum for the business analytics programming.
Yeah. So second question is that the idea of this whole PV program, you have told about technical aspects of PV, which is PV, Fabra, Aggregate, RMP, and those signals. What about non-technical aspects? Because I have seen certain documents like SDA or something like documents which wants the PV experience but does not have a technicality. Do you have any courses on that?
Exactly. So you didn't hear perhaps the last part of my previous when I was explaining. The program covers in detail the essential documents. So we understand the needs of the industry. So that program starts with the PSMF, very important. PSMF is we cover in detail. We then cover the pharmacovigilance quality management system, a development of a complete quality management system in PV. We cover then SOP writing, how the SOPs are written. Then there is a part called safety data exchange agreement. So how to document, how to author a safety data exchange agreement, how to review the safety data exchange agreement. And last but not least, the audits and inspections. We also cover that how to face audit if you are a vendor side, if you're a CRO side, how to face sponsor or a MAH audit, or how to face inspection eventually if you are in MH or even in a CRO.
And how to create audits.
Yeah, of course. There is Preetham, everything cannot be done in too detail in these programs, but we do have a program completely focused on PV audits and inspections. So that goes for almost 16 to 18 sessions of PV audits and inspections program. But in this program also, we touch the PV audits and inspections, but not obviously as detailed as what we will do in a full specialized program in PV audits.
Oh, no, because in the market also, I have seen certain programs which are audited.
Absolutely. So we have a very good program in PV audits and inspections.
And as you detailed up, it remembered me of GVP modules, because you started with PSMF is module 2. So all the things which you have detailed now, everything is related to all the I was just trying to remember
PQMS. I mean, if you want to relate the quality overall GXP, GVP point of view. So a lot of whatever things you're going to learn, PSMA point of view, PQMS, SOP writing, that's all part of quality, Preetam. So once you learn the quality, you are you're getting ready for the audits actually.
Thank you so much. Thank you.
Okay,
Uh,
thank you.
Hi, I'm Dr. Dikshita here. So I would like to know if any courses that are conducted by Dr. Shivakumar, like how do I enroll in the
Madam,
courses?
the program in the institute when we run, it's not run on the basis of one faculty. These are large programs. They run for about 6 months duration, and the faculty is mixed. We have the all senior level people like Dr. Shiva, like Gaurav sir. There are a lot of senior people who come and teach the program. So yes, to answer your question, Dr. Shiva will also be teaching the part of the program, and also Gaurav sir and other senior level trainers.
Okay, thank you.
Yeah, thank you. Anyone else? You have questions?
Some people have been asking on the questions, sir, that whether they'll be certified for this or whether the recording will be available.
Yeah, so the recordings will be available on YouTube. I think this will be going on YouTube, so you can all go and sort of see this session on YouTube. It will be available. No certification for this webinar. Any other questions you have? You want to unmute and ask? Be happy to answer if you have any other questions. And for all the freshers, Pallavi, you are asking what is the Gaurav sir, this is for you, targeted to you. What is the difference between DSP and medical review in point of okay. So from a DSP and medical review, drug safety physician, medical review is the same thing, Pallavi. It's for the medical doctors. So doctors, the drug safety physicians who do the medical review, who do the review of they are called drug safety physicians, or sometimes in different companies, they're called medical reviewers as well.
Yeah, so these are interchangeably used in the industry. DSP or as a medical reviewer, they are the same. The role is defined, it could be defined in terms of only working on the individual cases or working on the aggregate reports or working on the risk management plans, right? So that could be defined. However, these are the same.
Hello, sir.
Yeah. Yes, please.
I'm Dr. Pankaj Magar. Currently, I have 1.7 years of experience in medical review right now in ICSRs. I'm looking forward to learn aggregate reporting. In my department, I could see the aggregate reporting reports are being prepared by informed like authoring. So in this program, we also learn how to write the reports like as they are doing, because I could see doctors are only reviewing the aggregating reports, but they don't know how to author, how to write. So
yes, sir. That's Dr. Pankaj. Hello, how are you doing?
Hi, sir. How are you?
Good. Yes.
I'm good.
So yeah, Dr. Pankaj, it's a very important skill. So for doctors, also the medical doctors who are involved into the drug safety domain, they must understand, they must know, they must have experience of authoring the aggregate reports, because authoring will give you a different experience. Actually, the reviewing is great, but authoring will give you a complete deeper understanding of the subject actually.
So in this course, we will be
Yes,
authoring the report. So we will learn how to write a complete
live session to author, and you will be at the end of the program authoring one report, which will be PBRER. Will be giving you the the relevant cases, and then they will be asked to do participants will be asked to do the complete authoring of the report after they have gone through the various sections learned various sections, how to write various sections. End of the program will be write, author a report, which will be reviewed by the NCDA trainers.
Yes, and Pankaj, to answer you again, we will be having all these sessions for anyone who's a part. They will be writing all the sections. Okay, so once you write all the sections, I think you can review the document in a better way than just just looking at the medical section. It will not give you the complete picture. So yes, we will be looking at how to draft all the sections, and then eventually you'll be drafting one complete report.
Okay, thank you, sir. Thank you for this.
Right. There was a question from Pallavi. Do you do any hands-on the experience? Yeah, Pallavi. So the first part of the program, this starts with medical review. So we do two types of medical review. We do the clinical cases for the clinical trial cases medical review, and also for the post-marketing medical review. So yes, there will be practical exercises.
Any more questions?
Okay, if no more questions, you can always reach out to us. You can reach out. Does this program come under the purview of medical writing? No. Okay, so Dr. Anupam, the medical writing is a very vast term. It starts with the medical writing regulatory point of view. You do the CTDs, ECTD is kind of a medical writing. Then you have a pharma industry medical writing for the physicians, for the patient literature. Publication writing is a part of the medical writing industry. Then you have this medical writing part in this program is covered from a pharmacovigilance point of view. So all the aggregate reporting, which is a part of the medical writing, is covered in this program. At Cliniminds, we do offer a very specialized program in medical and scientific writing, which covers the all aspects of different types of medical writing. But in this particular course, we cover the PV medical writing, which is aggregate report writing.
Yeah, so this is again focused on the niche area. As Kamal sir was saying, this is focused on the pharmacovigilance domain. And so the medical writing here covers all the aspects of pharmacovigilance, whether it is the risk management plan or the aggregate reports, DSURs, or the PBRERs or the PADERs, right? So that is what we will be covering in our program.
So Rohit, typically a class duration is about anywhere between 2 hours, 15, 20 minutes, which includes the theory sessions, practice exercises, question answer sessions. So it's about 2 hours, 15 minutes. Generally, most sessions are about that. Okay, thank you, everyone. I think it's no more questions. So I'll let you go, everyone. You can enjoy your Sunday now. Thank you very much for joining the program. We will be in contact shortly with more information. Thank you, Gaurav. So thank you for your time today.
Thank you, sir. Thank you. Thank you, everyone. Thanks for joining, and look forward to seeing you all in the program.